My research is about generate Ponatinib resistant CML cell line models. So because Ponatinib is now used as a second-line therapy in CML, chronic myeloid leukemia, but we don't really know what the mechanism will cause Ponatinib resistance, and that's my research aiming to do, to investigate the resistant mechanisms, but in cell line models.
Okay. So how have you been modeling cell lines compared to human tissues?
I mean, the cell line is derived from human blood crisis cells, and at least they share, like in certain type of similar. It's not really the same as in human, but we can. Well, I mean, in previous publication, what we see from the cell line, the resistant mechanism, most often can imply into human. So I think it's quite like applicable. Yeah.
And are there any regions of genes that you've been working with especially?
So because BCR-ABL is a gene in CML that causes this disease, so obviously we will monitor the BCR-ABL1 gene like very regularly in my resistant line. And the other thing is we also monitor any gene got modified in my resistant line just to check what's the reason to cause resistance. And this one is called AXL. It's a receptor tyrosine kinase, and it was found to cause like AML, CML, and multiple other cancers. And what we find here in CML is this gene got overexpressed in the Ponatinib resistant cell lines, and it may be applied in human as well.
Okay. And when it comes to the results of this trial, figuring out the resistance, can you tell us more about that?
Okay, so I have generated 4 Ponatinib resistant cell lines. 2 of them has been exposed to the other TKIs before, so it has been exposed to dasatinib before Ponatinib, because Ponatinib is the second line. So when patient goes to Ponatinib, most of them have been exposed to the other TKI before. And in these 2 cell lines, BCR-ABL dependent resistant mechanisms are the main reason to cause resistance, and this including BCR-ABL overexpression or mutations, compound mutations in BCR-ABL. And I have another 2 cell lines that never been exposed to other TKIs before Ponatinib. And in these 2 cell lines, like I said before, the AXL overexpression is the reason to cause resistance in these 2 models.
Okay. Do you think these might be the leading drivers in resistance, or is there more yet to find?
I think, well, I'm quite confident with my result that in TKI naive setting, so BCR-ABL independent resistance, including AXL overexpression or other signaling overexpressed or downregulated, is the reason to cause resistance to Ponatinib. And in the setting of the cell line, or even maybe in patients that has been exposed to the other TKIs before Ponatinib, that compound mutation may actually lead to the resistance, if the resistance ever happen.
Do you know if the regions, the AXL, the BCR-ABL, have been implicated in other resistances beyond these TKIs?
Okay, so compound mutation is quite like it has been observed more frequently in the CML patients, especially when they have sequential TKI treatment, including imatinib, nilotinib, or dasatinib, and then Ponatinib. And BCR-ABL independent mechanism is, I think it's less, but has been observed before as well. So it's still a possible reason to cause resistance. And if you're talking about different disease other than CML, like maybe ALL, I think because Ponatinib and the other TKI also use in pH positive ALL, so similar mechanism will also lead to resistance in ALL.